Am I right?
Yes, sir.
Okay. So can you tell where exactly I have stopped?
So step two, using the probes.
Okay. So using the probe, right? So good. Okay. So let me continue from that. Okay. So I started with molecular docking study. Molecular docking is a kind of making complex between the protein and ligand molecule. You can actually make complex between two molecules or even more than two molecules. But normally we used to make complex between two molecules, protein and the drug-like molecule or the ligand molecule. Is my screen visible?
Yes, sir.
Yes, right. Okay, fine. Very first step is that we should solve the 3D structure of the drug target. Because molecular docking is a kind of structure-based drug design. So we should have the 3D structure of the drug target, and without having the 3D structure, we cannot proceed with the structure-based drug design. Another word, we cannot proceed with molecular docking studies. Okay. So very first step is we have to solve the structure somehow. The best possible way to solve the structure is going for X-ray crystallography. Other techniques also available, but normally in drug discovery they give importance for the X-ray crystallography method. So once if you have solved the 3D structure, right? So while solving the 3D structure, normally the crystallographer will try to solve along with some known inhibitor or the drug molecule, because if we do so, then it will be easy for us to identify where the binding site is present. But not always we can succeed in that process. There are some cases we may only able to crystallize the protein molecule, not along with the inhibitor. There are cases, right? So in that case, we have to locate where exactly the binding pocket is present, and we have to use the solvent taxable surface area to find out where the major binding pockets are present, and then we can narrow down which one could be the possible active binding pocket. So once if you know where the binding site is present, you will have very good idea about what are the amino acids which are lining that binding site. And each amino acid is made up of atoms, and each atom should be spherically represented as represented using the van der Waals radius. Okay, so here in this image, okay, if you missed the previous lecture, just recap, right? So each atom, okay, so this sphere, gray color sphere, or each atom which are lining up the binding site. Okay, so the radius of each atom is the van der Waals radius, right? So we can say this is this image, right? So the one on the right hand side, okay, that's surface image of the amino acids which are lining up the binding site, right? So second step is what we do is we have to take some probe molecule, probe atom. It can be either a atom or molecule. So if I say water, it's a probe, right? So water is a molecule. So we have to take a probe. For this case, the size of the sphere should be in the range of 1.4 to 1.5 angstrom. Okay, so if you look at the this range 1.4 to 1.5, we can say that is actually the size of the water molecule. So that's why we call this as running the water molecule. Literally what we do is we run the probe, right? So once if you run the probe on this surface, the probe atom will start rolling from one point to the another point. During this process, the probe atom will make contact with the van der Waals radius, the sphere that you have generated in the previous step. So these are the atoms which are lining up the binding site, to which to some atoms the probe atom is making contact, and that is traced here. Okay, so you can see this surface is kind of a convex surface, and here you can see this region is what is called as a re-entrant, which means that beyond this point, the probe atom, that's the water molecule, cannot enter. Okay, so this is a maximum to which it can make the contact. Right, so that region is what is called as re-entrant. Again, this is kind of re-entrant. Beyond this point, the probe atom cannot enter, right? So this is how we can define the binding site exactly. So moving to the third step. Okay, so now we know what is the 3D structure of the drug target. we have good idea about where the binding site is present, right? uh, we know, right? So we know where the binding site is present, but we have to dictate that information to the computer. So computer, we have to tell the computer, we have to train the computer in some meaningful way that this is my protein, and this is the area in which I have the binding site, or this is the area in which the protein has the binding site, right? uh, so then if we give this information clearly, then the algorithm can take the ligand molecule one by one, and they can fit it inside the binding site. Okay. uh, so that is what I have represented here. So the empty space of the binding site, what we identified in the previous step, has to be defined in a way that a computer program can understand before ligand can be inserted, right? So as a human, we can very easily understand, okay, this is the area where we have the binding pocket. But we have to train the computer in some way, in some meaningful way, that it can also understand that that is the place where the binding site is present, and that is the place where the ligand should be kept, right? So how normally they did in the UCS of doc algorithm is that very simple. Okay, so say for example, so this is what exactly we have identified. This region is what exactly we have identified in the previous step. Okay, so by rolling the probe water molecule, so we have identified that this region is exactly the binding site of this protein molecule. Hope it is understandable, right? So now we had to convey this information to the computer that this vacuum region, okay, this solvent accessible region, is the binding site where the ligand has to be kept. That is what we have to do, right? So that is what we had informed to the algorithm. How they did this in UCSF doc algorithm is that they took sphere, okay, of different size. Okay, so here if you look at carefully the blue color sphere, so not all the spheres are of same size. Do you agree? So different spheres having different size. Am I right?
Yes, sir.
Yes,
Okay,
sir.
so within this binding pocket, lot of spheres are inserted. So what I mean is, not all the spheres are having same size. Each sphere, it has its own size, right? So second point is that, so once if they keep the sphere inside, right? So they had ensured that each sphere that is kept inside the binding pocket is making contact exactly at 2 points of the binding site. Okay, so here I just give one example. Okay, if I take this sphere as example, this sphere is making contact at this position and at this position. So I can say that this sphere is making contact at two positions. Am I right?
Yes, sir.
So if I take this sphere, can you tell how many places it is making interaction within the binding site to the binding site amino acids or atoms? How many points it is making contact? So can you say it is making contact at this position?
Yes, sir.
Can I say it is making contact at this position? So how many total number of contacts?
Two.
Two, right? So now you just look at any sphere that is inside the binding site. Do you agree that all the sphere which are kept inside the binding site to fill the binding site area, they make contact only at two positions?
Yes, sir.
Yes, right. So if I take the third one, just for example, it's making contact at this position and this position, right? So this is how they define the binding site to the computer algorithm. This is a way how they make the computer to understand that This is the place where the binding site is present, and this is the volume of the binding site, and this is the exact place where the ligand molecule should be kept. Is it understandable?
Yes, sir.
Yes, right. Okay. So if you understand this, let me erase the annotation. Yeah. So what is the next step? The next step is, uh, uh, can you look at the image on the left hand side and the right hand side? Do you find any difference? So this is to my left hand side and this is to my right hand side. You just look at, compare the image which is present the left hand side and right hand side. What differences do you find? What major difference?
The chemical structure is fitted inside.
No, it just leave the chemical structure. You think that there is no chemical structure. Apart from that, what difference you find? Do you find any other difference?
Space is there. No, sir.
Hmm? So can I say that the kind of number of sphere is reduced in the right hand side image?
Yes, sir.
Is it clearly visible?
Yes, sir.
See here, you should carefully look at the screen. This sphere is missing in that. Do you agree?
Yes, sir.
Yes, sir.
Am I right? Okay. So but that is satisfied by this sphere. Do you agree? That area. Am I right? Okay, the
Yes, sir.
point is that after this step, I don't want to go in detail, but I just wanted to tell, right? So they reduce the number of sphere. There are different ways to that. They had ensured that even after reducing the number of sphere, so the binding site information is clearly dictated to the computer algorithm. That is what they have done in the right hand side image. Okay. So what is the next step here? You can see very clearly that the number of sphere is reduced to great extent. Do you agree? Am I right?
Yes, sir.
So some cases initially there may be two sphere here, okay, which are merged, right? But we can say the entire binding site is clearly dictated to the computer algorithm by means of this sphere, and this is called as the negative image of the binding site, and this information is the input for any docking algorithm as the binding pocket. This is defined now as the binding pocket to the computer with the help of the sphere, right? So let me erase the annotation for clarity, right? So after removing the unwanted spheres, once if you clearly defined the binding site, so what they do is they label each sphere. So here they have labeled as A, B, C, D, E, F, G. So each sphere, the centroid of each sphere, right? So is labeled with different alphabets like A, B, C, D, E, F, G. So depending upon the volume, right? So this number will the number of sphere will differ. Okay. So here, in this example, you had to assume that this molecule has the ligand molecule. Okay, so here, one second. The ligand is missing. Okay, okay. The ligand is here. This is the structure of the ligand molecule. You have to imagine, right? So because I don't have this in the next slide. Okay, so now I'm going to use two terms. One is this pair. Okay, so this is called as Okay, one second. So this ligand molecule is now converted into pseudo atom. Okay, so here this is now your ligand molecule. So normally, how do we represent the ligand molecule? How the ligand molecule is represented in this image? Can I say it is represented as lines? So you know the different types of representation, right? The simplest type of representation is line. We have ball and stick. Am I right? So I think you have used PyMol in some of your lab session. So small molecules can be represented as lines or ball and stick, spear. Am I right?
Yes,
So
sir.
here the representation of the ligand molecule is line. Okay. So now the same ligand molecule is represented using some spear, and these are what are called as pseudo atoms. Again, the previous image I have shown the ligand molecule as lines or stick. We had to convert that into pseudo atom. So which means that these are the various atoms of that ligand molecule. Okay, we're not taking the ligand molecule as line or stick. We convert that into pseudo atom. Is it understandable? So the centroid of this sphere is the centroid of the atom. So then we can number this pseudo atom. So for this ligand molecule, for this example, it goes like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. They have numbered the pseudo atom for the ligand molecule. So now they have labeled the spheres within the binding site. Can we measure what is the distance between A to B here? Centroid of A to B, B to C, A to F, F to G, F to E. That is what they have labeled, right? So we can measure the distance. Any combination we can measure the distance. So similarly, in any combination we can measure the distance between the pseudo atoms, centroid of the pseudo atoms like 1 to 2, 1 to 3, 2 to 3, 3 to 8, 3 to 9, 8 to 9, 1 to 4, and so on. Is it understandable?
Yes, sir.
Yes, right. See, if you don't understand, just let me know. I can give some other example. Okay, that's why I'm asking again and again. So if it is measurable, if the distance between 1 to 2, 1 to 3, 1 to 4, 1 to 5, and so on, if it is measurable, then can we create a distance matrix for this? You know what is meant by distance matrix, right? So similarly, we can create a distance matrix for the spheres which are present within the binding site. Am I right? So what we can do is we can compare the distance matrix of this and the distance matrix of this, and we can try to fit this molecule, this pseudo atom, inside the binding site, such a way that most of the pseudo atom whose centroid should be exactly fitted in the centroid of the spheres which are present within the binding site. Does it make sense? I cannot go to the next slide without getting any response. Is it understandable or not?
Yes, sir.
Okay. So that is what exactly this UCSF dock algorithm it does. Okay. So for fitting the ligand molecule inside the binding site, again I repeat, okay, conceptually very important. First, it fit different sphere inside the binding site, and it makes it label each sphere, and the input ligand molecule, whatever is the number of ligand molecule, it convert that ligand molecule as a pseudo atom, and it create distance matrix for each ligand molecule. As well, it creates distance matrix for the sphere which are present within the binding site. Then it compares the distance matrix of the ligand molecule and the distance matrix created for the binding site sphere, and it compare the distance matrix and try to fit the ligand molecule inside the binding site. Okay, that's what exactly it does. So if I show the next slide, to some extent you can understand. So can we say that the ligand molecule is fitted well in this image? Mhm. Can I say that the ligand molecule is well fitted inside the binding site?
Yes, sir.
Yes, right. So, but we cannot say that this ligand molecule is well fitted inside the binding site. Can you tell why? Because this is the area till the binding site is present, but one of the pseudo atom that is present outside the binding site. So we can say that there's a kind of steric clash here. So the algorithm will recognize that, okay, this is not the correct fit for this ligand molecule, and this is correct fit. Are you able to understand?
Yes, sir.
Yes, right. So that is how the ligand molecule is fitted inside the binding site. So again, I repeat that the same ligand molecule can be fitted inside the binding site in different orientation. We discussed about this in the previous lecture with a couple of examples. You have to remember. So the same ligand molecule in this image can be fitted inside the binding site in a very in different ways. Some of the orientation may be acceptable, and some of the orientation like this may not be acceptable. So whether it is acceptable or not, that depends on whether any pseudo atom is making any steric clashes or not. That is what is illustrated in this image clearly. So once if you fit this, the next step is that we have to calculate what is the energy or score for that fit. Again, you have to remember the previous lecture in which I talked about two important complementary. One is the shape complementary, another one is the stereoelectronic complementary. So if both shape complementary and stereoelectronic complementary, if it favor, then we can expect the molecule will be having a very high affinity. If the molecule has only shape complementary and lacks stereoelectronic complementary, or having a good stereoelectronic complementary but lacks shape complementary, either way, the affinity of the ligand molecule will be less. Okay, so since the same molecule can be fitted in different ways, there should be some method for ranking or calculating the or estimating the energy or the affinity of the ligand molecule and the protein molecule. Okay, for that, if you take any docking algorithm, the second part of the algorithm should be scoring part. The first part is docking. That is what I discussed so far. And the second part is the scoring. We have to score. We have to tell that which pose is the best one. So comparing 1 and 2, which pose is the best one? If I compare this and this, which one will have higher affinity?
One, sir.
Okay, one will have, absolutely right. So one will have much higher affinity comparing to the two, because of the steric clash which is present in the second conformation. So how do we say this molecule is having higher affinity than this? We cannot randomly tell that, and we should go for mathematical calculation part, and that is the scoring part. So what I'm showing here is the very simplest form of a scoring function. Okay, it's a kind of mathematical expression which has different terms, and all those terms present in this equation which contribute for the overall binding energy. Okay, so let me tell what is meant by binding energy. So if you have some protein molecule like this, say this is the binding site of the protein molecule, and this is your lead molecule. So once if you bind this, if this lead molecule binds to this protein molecule, so obviously some kind of interaction should take place between the ligand molecule and the protein. So whenever there is a formation of bond, some amount of energy should be released, and the amount of energy, okay, of this complex, that is given here as the delta G bind. Okay, so I just wanted to give what are the various terms which are represented here. So what is meant by delta? Okay, let me ask you a question. What is meant by delta G? This you might study in your 11th standard, 12th standard, and first year chemistry. What is delta G? It's an energy term which is very commonly used. Huh?
Gives free energy.
Free
Okay,
energy.
it is the Gibbs free energy. Absolutely right. It's Gibbs free energy. Okay. So here, everywhere, wherever you see the delta G, that is the Gibbs free energy, right? So delta G bind is the contribution of different energies of the system. Okay. So again, what I shown here is a very simple form of the scoring function. It's not complex one. Okay. So what is the delta G solvent? Okay. So say for example, you have a protein molecule, and you have say you have a ligand molecule, and your intention is you have to dock this ligand molecule inside the binding site of protein. Okay, so say both the protein molecule and ligand molecules are present within the cell. So we cannot directly bind this because the protein molecule naturally it is surrounded by some water, because the cell mainly made up of large quantity of water. So this ligand molecule can also be surrounded by the water. So if you want to bind this ligand molecule to the protein binding site, what we should do first? I wanted to bind this ligand molecule inside this protein. What should I do? Looking for some answer. I hope my slide is visible for you. Is the question understandable?
So first we have to find the binding site.
Though no, leave those stories are, we know where the binding site is present. For the given scenario, what should I do? I have a protein molecule which is surrounded by water. I have a ligand molecule which is also surrounded by water molecule. I just wanted to bind this ligand to the protein binding site. That's all. What should I do? Okay, say okay. I just okay. So this is the binding site. Assume that I know where the binding site is present. What should I do? I have to fit this ligand inside this binding site. The answer is very simple. At least someone can try to answer.
Near the protein, sir.
Sorry.
Keeping the ligand molecule near the protein.
Yes, madam. I can bring this ligand molecule closer to the protein. Okay, what should I do? Should I remove this water molecule first or not?
Yes, sir.
Okay, first what I do is I have to remove this water molecule which surrounds this protein, as well as I have to remove the water molecule that surrounds this ligand molecule. Okay, then what may happen is I can bring this ligand molecule closer to the protein. Am I right? I can fit this ligand molecule inside the binding site. So is it possible to fit without removing that water molecule?
No, sir.
No, right? So okay, this is called as the energy contribution by solvent. Okay, this is exactly called as a solvation. Always the protein and the ligand molecule will be surrounded by water molecule. It's and if you want to bind this ligand molecule inside the binding site of protein, the first step what should happen is the desolvation should take place. So this is in solvated state. So desolvation is we have to remove partially the water molecule surrounding the protein and the ligand molecule, and then we can fit the ligand inside the binding site. Okay, so for this process, energy is required or not? If you want to remove this water molecule partially, do we need any energy or not? I hope you studied the thermodynamics, right? So at some point of your 11th standard or 12th standard. So do we need energy for removing this water or not? I really don't know whether these questions are simple or difficult question. Even the 6th standard students, they can answer.
Sir,
Yeah.
sir, I have a doubt, sir.
Yes.
Sir, mostly the buried regions will be hydrophobic, no, sir. How water will be there, or
The
how
what I am
the
not talking about the buried region. Okay, what about the surface? I agree
sir,
with you. The buried region is hydrophobic. I agree with you that we cannot expect the water molecule to present inside the buried region. I'm not talking about the buried region. The
sir,
protein
there
is the outer region is it's hydrophobic or hydrophilic.
that is hydrophilic, sir. But
Okay.
ligands will be bound to the binding site only, not to the surface, no.
Can be okay. So you have a protein molecule, right? You have a protein molecule. The binding site is buried. You are saying that the site is buried. What does it mean? It is present somewhere interior. Do you agree?
Yes, sir.
Yes, right?
Yes,
But
sir.
the surface is the outer layer, right, of the protein. You have to think the protein in three-dimensional way. Am I right or wrong? Okay, so okay, I just want to clarify this. Maybe I take some good example, right? So I understand your question. One second. One second. I just
Okay, sir.
yeah. I'm giving some example. Wait. Okay, I just take some protein as example. Let's take is my screen visible?
Yes, sir.
What is on the screen? Are you able to see pi mol?
Yes,
Yes,
sir.
sir.
Yes, right. Okay, so I take this as an example, right? So it will be easy for you to understand. Okay, so here I have a protein molecule. Can I say that this region is the buried region, the one where you have some kind of fragment or ligand molecule?
Yes, sir.
Yes, right. So which means that that region is the place where the water molecule cannot interact. Am I right?
Yes, sir.
But do you agree that as a whole protein, right? So the surface of the protein is hydrophilic in nature. Am I right?
Yes, sir.
So the water molecule can completely surround the protein, but it cannot enter deep into the buried region. Do you agree?
Yes, sir.
So if you want to fit the ligand molecule inside inside the binding pocket, even in this example, you can see there is one water molecule within the buried region. Are you able to see?
Yes, sir.
Okay, so which means that the protein is completely going to get surrounded by the water molecule, but very few water molecules they can get enter into the hydrophobic pocket. If that is the case, if you want to fit the ligand molecule inside the binding site, the first thing what we have to do is we have to remove the water molecule that surrounds the binding site. Does it make sense?
Yes, sir. Okay, sir.
Are you able to understand?
Yes, sir.
I don't know whether my explanation is convincing or not.
I understood, sir. Thank you.
You understood, right? Yeah, okay. So that is what I have illustrated here. Okay, whenever I talk about any protein molecule, you have to think the protein as a three-dimensional, in three-dimensional way, right? So that is what I have given here. So do you people now agree that we have to remove this solvent for the ligand molecule to get enter into the binding site? The one I have shown in this. Am I right or wrong? So the water molecule is going to get surrounded here, and we have to remove few of those water molecule to give path for the ligand to get enter into the binding site. Do you agree? Okay, I assume that you people understand, right? So I don't want to take too much time asking again and again. So that process is called as desolvation, and there is we have to spend some energy. We, in the sense, the system, the protein ligand and the cell, they as a whole, the system has to spend some amount of energy to remove those water molecules from those sites, and the energy that is associated with that process is called as delta G solvent. Is it understandable?
Yes, sir.
Yes, right. So okay, fine. So again, we'll try to understand the next parameter which contributes next energy next factor that contributes for the overall delta G binding. The next one is called as a delta G conf. Okay, this may be new for you. You might not studied about this in any other coursework. So let me tell what is meant by delta G conf. So delta G, this conf refers to conformational. This refers to conformational energy penalty. You may note it down. This refers to conformational energy penalty. So I just want to give one example for you to understand what exactly is the meaning of conformational energy penalty. So for example, you have a protein binding site, and the shape of the protein binding site is something like this. Okay, this is the shape of the protein binding site. Okay, so this region is the shape of the protein binding site. So now you have a ligand molecule. So initially the ligand molecule is present like this, and one of the conformation of this ligand molecule is like this. Okay, both are same. So the A and B both are same, just graphical illustration for understanding the concept. The only difference between A and B is the A is present in one conformation and the B is present in another conformation. We already discussed about conformation, why it arises, everything when we talk about the importance of stereochemistry in drug design. So I hope you completed your molecular modeling 1 at least, in which you might studied about a term called as global energy minimum. Am I right?
Yes, sir.
So if you draw the potential energy surface graph, energy on the y-axis and angle of rotation AOR on the x-axis, so you will get some plot like this. It will differ from one molecule to another molecule, also differs from angle of rotation, right? So in this, can I say that this point is the global energy minimum conformation? So can I say that this point is the global energy minimum conformation?
Yes, sir.
Yes, sir.
So how do we say that? Because that is a point in the potential energy surface graph which has the least energy. Hope everyone agree with that. So I'm going to label this as global energy minimum conformation. Okay, so can I say this is local minima, this point? Can I say that this point is a local minima, local energy minima?
Yes, sir.
Yes, right. So can I say that local energy minima is also one of the stable state?
Yes, sir.
Yes, right. So I'm going to say this as local energy minima. You know what's the difference, right? So conformation is the different. So this is plotted for the same molecule but having different conformation by rotating the carbon-carbon single bond. Okay, so that's how we generate this potential energy surface. So I'm stating that this is A and B both are same molecule. A is present in one of its conformation, B is present in another conformation. So I am stating that A is global energy minimum and B is kind of local energy minimum. So which one will be having lower energy? A or B?
Yes, sir.
A will be having low energy comparing to B. But which of this conformation is bioactive conformation? Let me define what is meant by bioactive conformation. Bioactive conformation is the conformation of the molecule which can fitted in the binding site of the protein to initiate the biological response. Again, I repeat, bioactive conformation is the conformation of the ligand molecule that can exactly fit inside the binding site of the protein molecule, which can initiate the biological response. If this is the definition for bioactive conformation, in this example, A, B, which of this conformation we can call it as bioactive conformation?
B, sir.
Okay, so we can say that B as the bioactive conformation. Even though A is the global energy minimum conformation, it cannot produce the biological response. Only the conformation B, it can produce the biological response. Now the energy difference between this B and A, that is what is called as conformational energy penalty. So what does it mean? It means that, so if you have 100 molecules of A, if you have 100 molecules, most of the molecules will present in the conformation A because it is the lowest energy. Few molecules can be present in another conformations like B, C, whatever it is. But unfortunately, for this example, the global energy minimum conformation is not the bioactive conformation. It cannot goes and bind to the binding site to produce the biological response. The conformation B, even though it is slightly having higher energy, it can go and bind to the protein binding site to initiate the biological response. So there is a kind of energy factor which is involved in this. So if you want to convert the molecule in its conformation A to B, then the system has to utilize some amount of energy, and the amount of energy that is involved, or the energy difference that is between the conformation B and conformation A, that is what is called as delta G conf, or conformational energy penalty. Is it understandable? Does it make sense?
Yes, sir.
Okay, so let me erase the annotation, right? So I have clarified what is meant by delta G solvent, what is meant by delta G conf, right? So the third parameter in this simple scoring function is delta G int.
Sir, we can't hear you.
Is my screen visible?
Yes, sir.
I actually got disconnected. I don't know when I got disconnected. Can you tell exactly what I discussed last? Is delta G int understandable?
Yes, sir.
Okay, I don't know when exactly I got disconnected. I hope that you are connected till I explained the delta G int. So the next three parameters are very simple, right? So these are this you might study in your physics in your 11th standard or 12th standard. So the delta G int is just now what I explained. So delta G ROT, delta G TR, and delta G vibe, right? So delta G ROT is called as rotational. So the molecule will be in constant rotation, and this delta G vibe is vibration. The molecules will be in constant vibration, and this delta G TR called as translational rotation, translation and rotation. Okay, so if you take any small molecule, they will be constantly undergo kind of rotation they will constantly undergo kind of vibration, translational rotation. These are parameters which are associated with energy of the molecule. Whenever the molecule undergo rotation or vibration or translation rotation, there is some amount of energy which is associated, and those energy also contribute for this delta G bind. And if you want to calculate or estimate the delta G binding, the way to do is you can use simple contribution like delta G solvent plus delta G conf plus delta G int plus delta G rotational plus delta G translational rotational plus delta G vibration. So if you sum all this energy contribution, so finally we can say that is delta G binding. But that's not going to be the exact delta G binding. We don't have any method for exactly estimating what is the delta G binding. To some extent we can approximate that could be the delta G bind, right? So whatever I have shown here is a very simple scoring function, but don't think that the software will also use a similar kind of simple scoring function. Okay, so this is the scoring function, basic scoring function which is used in Autodoc. You can see the expression is completely changed, right? So in the previous slide I have given a very simple example, but now each term is expanded to a great extent, right? So many parameters which are included. So what I'm trying to say is that if you take any commercial or very standard molecular docking software, they may use a very complex scoring function. It is not essential that we had to understand all the terms which are present, but we have to understand that the expression, the mathematical expression for calculating the energy for scoring which pose is the best pose, that is going to be very, very, very complex. And this is exactly the scoring function which is used in Autodoc. Okay, so this is how we calculate the energy of protein ligand complex, and we rank which pose is the best pose. And that is all about the molecular docking and scoring function. Any question I can address? I think time is up. Any question I can address? Any question I can address? When is the next quiz?
Tomorrow, sir.
Tomorrow is Tuesday, Wednesday. No, not tomorrow. We will make it on Friday. Tomorrow we'll have normal regular lecture. Okay.
Okay, sir.
Okay, Friday is the examination, the second half, right? This time I'll go for some simple multiple choice questions. Okay. Is it fine?
Yes, sir.
Yeah.
Yes,
Yes,
sir.
Because in the first examination it is not tough. This is it's not related to mark or something. Just I wanted to expose you that what type of questions if you go for core company they may ask. That's why I put like that. Not all the quizzes will be of tough, right? So maybe next quiz will be completely of multiple choice question. You can easily answer. There won't be any issues, right? But the quiz is not tomorrow. Tomorrow we'll have the normal regular class continuation of today's lecture, and Friday we'll have the next quiz. Inform to your friends those who not yet joined for the session. Okay. Shall we wind up? Any question I can address in the next class?
Okay, sir.
Yeah. Okay.